Thank you very much. Well, good morning, everyone. It's a pleasure today to introduce grand speaker, Dr. David Hill, MD, PhD, Assistant Professor of Pediatric at the Children's Hospital of Philadelphia. Dr. Hill earned his MD, PhD from the Perelman School of Medicine at the University of Pennsylvania, where his research with Dr. David Artis focused on mucosal immunology. He completed his pediatric residency and allergy immunology fellowship at CHOP alongside a postdoctoral fellowship in immunometabolism under the mentorship of Dr. Mitch Laser. Dr. Hill is a nationally recognized expert in EOE, food allergy, and anaphylaxis, with numerous publications in very high impact journals. And today, he will present how clinical epidemiology can inform mechanistic immunology, both in patient and model system. It's a framework with broad relevance across immune-mediated disease. Thank you so much.
Thank you, Dr. Guirnez. Thank you all for being here. I really appreciate it. I'm very thankful to Dr. Guirnez, Dr. Lewis, Elena, and the Department of Pediatrics for the invitation and for organizing this visit. I've had a lovely time so far. I'm really looking forward to meeting with everybody today. So that introduction was very generous and wonderful. Thank you, Dr. Guirnez. I do hope that the paradigm that I'm presenting today will have relevance to pediatric diseases broadly, not only those related to the immune system. I do have some educational goals that were provided to you ahead of time, and so I will not read them to you here. Beyond saying, I hope that by presenting this talk in 3 parts, I'll walk you through how the use of clinical epidemiology can really be a driver for developing hypotheses that can then be studied through mechanistic mouse models as well as human systems, and that ultimately we can translate this knowledge into clinical innovation. I do have some disclosures. At CHOP's insistence, I did submit 2 patents on some of these technologies, and so I want to be sure to acknowledge that here. Otherwise, my lab is funded by various foundations as well as the NIH who have supported this research. I do not have any financial relationships with any biomedical companies or pharmaceutical companies that are relevant to what I'm going to be speaking about today. All right, so this story actually started with a patient that I saw when I was a senior medical student a few years ago. And this was a very interesting patient because this child had food allergy. However, there were aspects of the clinical presentation which were somewhat novel to us and previously not well understood. So this patient actually started with anaphylactic food allergy, the classic form of food allergy that affects somewhere between 4% and 8% of children in the United States. And being an allergist, of course, we performed our skin testing. And sorry, let me just find my pointer here. See if it's this one. No, this one. Oh, I see. There it is. All right. A little bit unwieldy. All right, I'm just going to leave it there. So on the y-axis on the left-hand side, you can see the skin prick test size. Initially, it was actually quite large, fitting with the symptoms presenting as anaphylactic food allergy. And you can see that over time, the skin prick test size reduced, consistent with potentially outgrowing that allergy. And we actually performed a food challenge, which the child passed. The reason this particular case was useful for understanding the connection between these 2 diseases is this child happened to have an endoscopy because of GI symptoms, and that endoscopy was normal. So you can see on the left-hand side, the esophageal histology there, which showed the absence of any eosinophils. So milk was reintroduced into the child's diet. The child tolerated that very well. And over the course of several months, up to a year, the child started to develop symptoms of another disease, another form of food allergy called eosinophilic esophagitis. The symptoms of that condition in young children are feeding difficulty, symptoms similar to reflux, abdominal pain, and ultimately it can actually predispose to failure to thrive and other sort of systematic complications. As a result of those symptoms, the child underwent a subsequent endoscopy, and you can see that in the middle image there, that indeed there were many eosinophils present, consistent with the diagnosis of EOE. And at that point, milk was eliminated and the disease resolved. Now, the reason this was a somewhat unique presentation is previous to this, our understanding of these two food allergies was that they were actually quite distinct processes, right? Anaphylactic food allergy mediated by IgE, EOE mediated by T cells. And it wasn't well understood that these two conditions could actually have a relationship to one another. So let me tell you a little bit more about EOE. The incidence is somewhere around 1 in 1,000 to 2,000 individuals. As I mentioned, it is a T cell mediated chronic food allergy. So the presentation actually is months to years. And oftentimes these children won't present to GI or allergy until 2 or 3 years of age or even later. It is a cause of esophageal inflammation. The eosinophils are the sine qua non, or the sort of the classic immunologic feature of EOE. However, we now know that there are many immune cells that help to mediate this disease, in some cases may play a more central role, for example, the T cells. And then, of course, in addition to the initial presenting symptoms, it can predispose to malnutrition, anxiety, and eating disorders. A critical feature of EOE that I'd like you to just keep in your mind is that for the majority of children, it is IgE independent. And the reason that's really important for this talk is our existing allergy testing modalities, whether it be skin prick testing or serum IgE levels, are largely not helpful in trying to identify EOE causal foods. And so this leaves a very real question and a very real knowledge gap in the field. And that is, how could we develop diagnostics? But in order to do so, I first wanted to ask, what is the relationship between anaphylactic food allergy and EOE? And the way that I first sought to do that was using clinical epidemiology. Now, full disclosure, I'm not a clinical epidemiologist. My PhD is in mechanistic immunobiology. But when I was a resident and subsequently a fellow, it became very challenging, as I hope you all can understand, to actually perform that kind of research, understandably. And so this was a tool that I saw being used very effectively and that I wanted to learn about. And so I teamed up with my South Philadelphia primary care clinic preceptor, who happened to be the head of informatics at CHOP, to learn a little bit about this technology. I don't consider myself an expert. It is something that I've done a fair bit of at this stage, and I'm always looking for experts to collaborate with in this regard. So this is a paper that is now almost a decade old, and it describes what at the time was a relatively novel approach to using EMR data, at least in the United States of America. And what we did is we took all of the data in CHOP's EMR that was made up of patients that had been seen at 4 urban primary care centers, 27 suburban centers, 16 specialty centers, et cetera, et cetera. And prior studies had actually shown that this cohort was nationally representative, in particular for urban and suburban patient communities. We then defined what we call an EHR-based virtual birth cohort. And the way that we did that is we used an open cohort design model. So all patients that had been seen at CHOP between, at the time, 2001 and 2020. And that included almost 180,000 children who, firstly, had established care before 1 year of life. So in that regard, it was an early introduction into the cohort. And secondly, had been observed for at least 2 years. Now, actually, the mean observation time for this cohort is closer to 5 years. So we actually have a very large longitudinal observational window for patients in this cohort. We then used a combination of ICD codes, food allergen data, and medication prescriptions to identify the onset of disease longitudinally across these children. And at this point, we had the tool that we could use to ask, what is the clinical relationship between anaphylactic food allergy and EOE? And so this information was reported in 2 papers in 2017 and 2018. And I'm going to quickly just give you the highlights for the sake of background. The first paper showed that a history of anaphylactic food allergy to either milk or egg did positively associate with an EOE diagnosis. In fact, the prevalence of EOE in children with a history of anaphylactic food allergy was about 10 times higher than that in the general population. In addition, we found that the most common foods that cause anaphylactic food allergy and EOE were actually shared. And so that was really important because it started us thinking that on a mechanistic level, that there could actually be shared immunology between these two conditions. The second paper did a more comprehensive analysis of the relationship between EOE and the other allergic manifestations. So what I'm showing you here is a model that's near and dear to my heart. It's both a clinical epidemiologic model as well as a mechanistic model of the relationship between allergic conditions. It's called the allergic march. It's not a perfect model, but it's one that I think is useful in terms of these kinds of studies and trying to understand clinical relationships. And so on this plot, I've shown you 5 different allergic conditions, starting with atopic dermatitis, then anaphylactic food allergy, asthma, allergic rhinitis, and finally EOE. And what you're seeing there is essentially the incidence of those conditions over time between 1 and 5 years of life. And once we had defined that relationship, we could then ask, to what extent does one condition increase the risk of developing a second? And the way that you read the graph behind me on the right-hand side is the primary diagnosis is on the left. So you can see the primary diagnoses from top to bottom, atopic dermatitis down to allergic rhinitis. And then the secondary diagnosis is across the top. And so you can actually see that all of the major allergic manifestations actually increase risk of a child going on to develop a second. So that is actually sort of a core principle of the allergic march, if you will. Many people in the room and in the audience might be familiar with the strong relationship between atopic dermatitis as a risk factor for developing anaphylactic food allergy, for example. So you can see that risk measured here at somewhere around 2.5 fold risk over the general population. What really stood out to us, however, was the very strong risk relationship between anaphylactic food allergy and EOE. And this was not previously appreciated, but you can see that a child with anaphylactic food allergy actually has a 9-fold higher risk of going on to develop EOE than a child without anaphylactic food allergy. And so this was really starting to underscore that not only is there utility in understanding the clinical relationship here, right, in terms of risk stratification, prognosis, but in addition that there could be shared immunology underpinning these two conditions. And so at that stage, I had completed my residency and my fellowship. I had actually completed my postdoc and was starting my lab. And I was in the position where I could actually ask to what extent was there shared mechanism underpinning these two diseases. And so that moves us to the second phase of our talk. This is actually a very good friend of mine, Amanda Muir. Does anybody recognize her? She's a GI investigator and physician at the Children's Hospital. Also focuses on EOE. All right, so what is the immunologic relationship between these two conditions? So for the non-immunologists in the room, I have what I consider to be a simplified diagram of the immune response to foods. You can see the purple cell in the top left corner. That is the dendritic cell or antigen presenting cell. It's taking up antigen from the environment, presenting it to a naive T cell. And that naive T cell has one of 3 fate decisions that it can then pursue. It can become a T follicular helper cell, which goes to the follicle and interacts with B cells. It can become a memory cell, and it can become a Th2 effector cell. So in the case of anaphylactic food allergy, of course, it's the top pathway predominantly, right? You have development of the follicular T cell. It goes to the follicle, it interacts with the B cell, it instructs that B cell to make IgE, and that's how you end up with anaphylaxis. In the case of EOE, however, as I've mentioned to you and a few decades of research suggest, it's the T cell that's really critical in terms of mediating that disease process. It's a chronic disease. There's lots of T cell infiltration, lots of T cell effector molecules, and subsequently T cell mediated inflammation. And so the question that we wanted to ask that was sort of a fundamental one in the question in the field that hadn't been answered is, can we actually detect food specificity in the T cell compartment? People had done this in anaphylactic food allergy, certainly. But in EOE, we had animal models, we had some suggestions in humans that it was the case, but we had never actually identified food-specific T cells in patients. And so we sought to do that. This was a paper from 2021 by an incredible research technician in my lab who is still with me. Her name is Julie. She's absolutely phenomenal. And what Julie did is she took a cohort of patients with food allergy, specifically EOE, And we knew in this cohort that the EOE was caused by milk. So we went back through our charts. We found a group of kids with EOE milk allergy. We recruited them. And then we recruited a bunch of controls. The controls were both non-allergic people, and they were people who had other forms of allergy. And we even had some kids who had EOE not caused by milk, right? So that we sort of covered all of the various options. Julie then isolated the peripheral blood mononuclear cells, stimulated them in vitro or ex vivo with purified milk proteins, and then looked at the immunologic response. And what Julie found was that compared to an unstim control on the left-hand side, a milk stimulation had minimal effect in a control subject along the top, but resulted in a dramatic and significant upregulation of IL-4 production in the T cell compartment as a result of milk stimulation. Now you'll notice in the middle there that we have a tetanus column. That's our positive control. All of these children were immunized to tetanus, and we could see a tetanus response, and we continue to do so and use that in our work as a positive control. And then you can see the cohort data normalized, on the right-hand side graph so that you can see all the data points there. So indeed, we found a milk-specific T cell response in children with EOE. And notably, we actually found it in the circulation. So we didn't have to go to the mucosa. We didn't have to use a biopsy. It appears that there are circulating food-specific T cells in children with this condition. And so that was very, very interesting. But we wanted to delve a little bit more into the molecular mechanisms by which recognition of food antigen was occurring in this disease. And so to do so, we used the exact same experimental pipeline, cultured PBMCs from a single individual. This was an individual with classic EOE. So they had been diagnosed with EOE to milk when they were very young. They removed milk, their disease resolved. They then had an accidental exposure. Symptoms came back. They were biopsied again. EOE was back. They subsequently removed milk. It resolved. So this is sort of your best case scenario child with EOE. And what we did is we took PBMCs from that child and we split them into two arms. We had a control arm that got no stimulation, and then we had an experimental arm that was stimulated with purified milk proteins. And at the end of a couple of days, we isolated the memory T cells by bead purification and then performed single cell RNA sequencing with linked paired TCR sequencing. So what that means is that for each individual cell, not only did we know the transcriptional information, the transcriptome, but we also knew not only the TCR genes that it was expressing, but the TCR clonotype, the combination of the two chains that make up the TCR, which mediate specificity, essentially, in that T cell. And so the first question we sought to ask was there clonal expansion, right? If this is a T cell mediated disease, we would expect that upon stimulation with the cognate antigen, that there'd be clonal expansion of those T cells. And indeed, we saw that. So this plot on the bottom left-hand corner, those light blue dots are the cells that actually showed clonal expansion with milk stimulation. To show that another way, here I've just graphed the milk on the y-axis and the unstim on the x, and you can see that there's more expanded T cells in the top left portion than in the bottom right. So indeed, there was clonal expansion of these T cells consistent with an allergen-specific T cell response. Now, if we flip back over to the RNA-seq, the power here was we could then ask, what is the transcriptional phenotype of those expanded cells? And what we expected is that they would show a predominance towards an allergic phenotype or a Th2 immune response. And that's what we saw. So on the far right-hand side of that third plot, in the light blue, you can see that there's a significant elevation in a sort of characteristic set of allergy genes or Th2 genes in the milk expanded T cell clones. And so this was very exciting. It sort of confirmed sort of a couple decades of research, both in animal models and humans, suggesting that there was milk specificity in the T cell compartment and that was mediating a Th2 cell response. But the utility of this data set actually went further. And that came from the fact that because we had single cell data, both the TCR clonotype and the transcriptional data, we could then look at the expanded clones and try to understand the extent to which they were expressing Th2 genes and whether we wanted to study them further. So what you can see in this last plot here is the 20 most expanded clones in the milk arm compared to the unstem. And then what we've done is we've graphed the Th2 gene signature on the y-axis. So you can see about half of these clones that expanded expressed high Th2 genes, and the other half did not. For the sake of the discussion today, I'm going to be focusing on the Th2 high genes because our initial goal was to clone and characterize a milk-specific pathogenic effector T cell. However, we're very interested actually in the Th2 low expanded clones as well, where they express a regulatory phenotype or potentially a different sort of effector phenotype. And so we're looking into that also. So for the sake of time, I will short circuit about a year of work. We looked at many of these clones for specificity, but for the remainder of the talk, I'm going to focus on clone number 4, which we found was indeed a milk-specific clone. So to remind everybody, up until this point, we have evidence that there's milk-activated T cell proliferation in the periphery. We have evidence that there's milk-driven clonal expansion of T cells, but we haven't actually proven milk specificity yet. To do that, you need to use a pipeline similar to this. And so what we did is we took the information related to TCR4, and we expressed that TCR in naive T cells. These were naive T cells from a volunteer. This person had no allergy, certainly didn't have EOE. We isolated their naive T cells and used a lentivector transfection system to essentially express our TCRs of interest. And at that point, we could culture with various antigens and look for a T cell response. So here I'm showing you evidence that indeed this TCR was milk specific. On the left-hand side is our control. This is the T cell culture with monocyte-derived DCs alone, no antigen. You wouldn't expect a proliferative response. The way you read this plot is the CFSE plot. So the x-axis is actually showing expression of a fluorescent dye. And then as a T cell proliferates, that fluorescent dye divides. And so you can actually see the sort of generations, if you will, of proliferation moving from right to left across the plot. The middle graph there is a control where we just stimulated our T cells with beads. And then the right-hand side is the data where we pulsed the monocyte-derived DCs with synthetic milk protein and then cultured them. Here I'm showing you the data for beta-casein. That is the milk protein that this TCR is specific to. I'm not showing you the data that it wasn't reactive to other milk proteins like beta-lactalbumin, soy, tetanus toxoid, completely unrelated proteins. Now, within the caseins, there's actually a fair bit of homology. So there's 3 major caseins in milk, and they share somewhere around 40 to 60% protein homology. And so initially, we weren't sure which of the caseins this particular TCR was specific for. The way we started to figure that out was using a limiting dilution. So here, the higher concentration is on the right, and we have alpha casein, beta casein, and kappa casein. And then we do a limiting dilution as you move to the left, and you can see that the TCR remains reactive to beta casein even at very, very low concentrations of antigen, whereas eventually you can dilute out the effect to the other casein proteins. So at this point, we knew that this was a beta-casein specific TCR, but we wanted to know the actual antigen itself because that information is actually critically useful in trying to design both diagnostics and potentially therapeutic interventions. We went about this 2 ways. Initially, we used a computational program that predicted the antigenicity within the protein itself, and you can see that data on the left-hand side here. We actually repeated all of this with an overlapping peptide library subsequently and confirmed it. But initially, we synthesized these 3 peptides, and then you can see here that peptide 1 resulted in specific activation of the transfected T cells, whereas the other peptides did not. And so this is the actual antigenic peptide. So as I mentioned, after this initial computational approach, we generated an overlapping library that spanned the entire of the beta-casein protein, and we tested all of those overlapping peptides, and we were able to narrow down the antigen to that amino acid sequence. And then in the red there is the predicted antigen itself from the binding prediction on the left. Now, what's really interesting is that this antigen, it's not the first time it's come up in the literature. So this antigen was actually reported out of Benaroya Research Institute about 10 years ago, but in anaphylactic food allergy. So finally, after essentially what had been 4 years of research, we were starting to see links back to our clinical epidemiology. And that in fact, at least in the case of beta-casein, there is evidence of shared antigenicity between these two, what we previously thought to be distinct forms of food allergy. All right, so the final piece of the puzzle here was actually the HLA restriction. And this was really important because if you're going to design something like a tetramer, a novel diagnostic, if you will, knowing the HLA restriction of the TCR is really important. So we did this using sort of a classic approach where we incubated these cultures with blocking antibodies that interfered with the interaction between the HLA and the TCR itself. You can see in the control condition here, when we stimulate our transfected T cells with monocytes pulsed with beta-casein, you get proliferation. And then when we add our blocking antibodies, you lose that proliferation specifically with anti-DR. So at this point, we had restricted EOE TCR4 to HLA-DR. HLA-DR was the molecule that was presenting the antigen to EOE TCR4. At this point, we really didn't know whether or not DR was relevant just to this TCR or whether it was more broadly relevant to the disease itself. So we actually went back to the original subject that we did all this work on. We asked for another blood sample, and we performed just a ex vivo stimulation in the presence or absence of anti-DR. And that's what I'm showing you on the bottom left here. And what was really actually surprising, and you can see it in the bottom right, when we incubated with anti-DR, we lost the majority of the proliferative response to milk. So what this tells us is that sort of two options. I think one option is less likely, and that is that all of this child's EOE is mediated by this TCR. I think that's less likely. I think likely there's multiple TCRs that are specific to milk or other foods in each child with EOE. The other option is that there's sort of a collection of TCRs, but they're all restricted to DR. I think that's potentially more likely, and it's actually very clinically relevant. Through some additional work and process of elimination, we were actually able to restrict this TCR further and show that it's restricted not only to DR, but DRB1 0701. So a very high level of restriction for this particular TCR. All right. And so with that, I'm just going to move into the final stage of my talk. And it's really the most important part, right? We've done a lot of work related to understanding these two conditions. How can we use that information to actually advance pediatric care? And so I'm just going to come back for a moment to this diagram to remind everybody that we've now shown that there are indeed antigen-specific milk responses by T cells in EOE. Importantly, we can actually detect them in the circulation. So these are circulating memory T cells. And that opens up right away the potential for the development of minimally invasive diagnostic. Okay, and this is my reminder just to remind you that our current diagnostics are not useful in EOE, unfortunately, because it is an IgE independent process. However, if we're able to detect the food antigen specific or food antigen activated T cells in the circulation, then that very well could be used for a diagnostic. Moreover, if we know information about the TCRs, the antigen epitopes, and the HLA restrictions, that information is actually very important, not just to develop biomarkers for disease, but also potentially prognostic modalities. And so how can we use this information to drive clinical innovation? So this is an assay that we've been developing in my lab. We initially did it for milk based on sort of the information I just showed you. We've now transitioned the milk test, if you will, over to our clinical immunology lab, and they're developing it internally, and hopefully we'll have it available in 2025. Meanwhile, my lab is repeating this process for some of the other food antigens. And the way that we do the test is actually very similar to our experimental procedure. We isolate PBMCs from the patient. We then stimulate those PBMCs with milk proteins along with various controls. And we look for activation and proliferation by CFSE dilution after 6 days. This was the initial cohort that showed that this modality could be useful. It was 34 children with and without EOE. And when we looked at the data from this cohort, we found that indeed there was a significant enrichment in milk-activated T cell responses in children with milk EOE. And moreover, if you back calculated the potential sensitivity and specificity for this approach, it was somewhere in the range of 88 and 82%. So pretty good, but a relatively small cohort. And importantly, this was a retrospective cohort, right? I knew a priori what kids I was enrolling and what controls. Now, we did everything blinded subsequently from that. But what we really wanted to show, and importantly, if this was going to be a diagnostic, was that this approach could actually predict the development of EOE milk allergy. And so over the last 3 years, we've been doing a prospective observational study. It has a real-world design where we enrolled subjects with symptoms of EOE who were undergoing endoscopy and biopsy and had unknown milk status. We then performed and interpreted our assay a priori, and then subsequently interpreted the esophageal biopsy. About half of those kids in our practice are diagnosed with EOE, and the other half do not have EOE. And we then followed the EOE group over time to see whether or not they'd be diagnosed with milk as a causal allergen. I know there's people in the audience saying, why didn't you do an interventional trial? This was a prerequisite for us doing an interventional trial. So we're starting that now, but we first had to establish, rightfully so, to our IRB, that this was even useful at all. So initially we used an observational design. This is the enrollment data. So we started with 211 subjects. It was a minimal risk trial. And so we basically got whatever blood was left over at the end of a series of blood draws. And so we did have various exclusions along the way related to insufficient amount of blood. We also had clinical exclusions, right? This is an observational trial. So many of these kids, disease resolved with PPI, or they elected not to undergo diet elimination therapy. They went on dupilumab, right? And I had no control over that. But ultimately, we were left with 22 subjects without EOE that were our controls, and 37 subjects with EOE. And then you can see the breakdown of the clinical outcomes of those EOE subjects below. But the important thing was that in all comers, so these are either controls compared to children with EOE milk allergy, we did see a significant enrichment in the outcome of milk-specific T cell proliferation. And that equated to a sensitivity and specificity of 92 and 82%. So actually a little bit better than our retrospective trial. And I was certainly pleased to see that. But I think the real power of this trial was when we didn't look at all comers, all subjects, but instead just focused on the kids that we knew had EOE, right? Because we have the biopsy. So we know which ones ultimately were diagnosed with EOE. Within that group, the performance characteristics were actually much higher. So approaching 92% sensitive and 100% specific. And so this is really informative because it means that the initial use case here for this diagnostic or biomarker, might be after the initial endoscopy to help to design a sort of personalized elimination diet, if you will. Now, there's other potential approaches for this information. So one of them is a tetramer-based approach, right? The current assay is essentially a functional T cell assay. We have to culture the T cells ex vivo for 6 days. It works, but it's somewhat cumbersome in the laboratory. And so you can imagine that an approach where we can detect the T cells directly ex vivo could be superior. And thankfully, in the case of EOE TCR4, we had the information available to actually try to make a tetramer-based diagnostic. For those who aren't familiar, a tetramer is essentially a synthetic MHC molecule with the antigen bound to it. And then you can use that kind of like a flow cytometry antibody to go in and find specific T cells. And so this is a sort of proof of principle, if you will, that indeed in our transduced T cells, tetramer works very well. Has a very high degree of correlation, essentially 1-to-1, with expression of the TCR itself, which we can measure via a different antibody. And then in addition, I mentioned the HLA data could be useful from a prognostic point of view. And so HLA-DR0701 is the HLA that this particular TCR is restricted to. I've also shown you evidence that in this patient, DR seems to be mediating the majority of presentation of milk antigen. And it turns out that 0701 is actually, it is a common HLA allele in Caucasian individuals, but it also is significantly enriched. It's about twice as prevalent in children with EOE milk allergy specifically. So this opens up the potential for akin to celiac disease, right, where you could essentially have a screen of multiple HLA alleles and try to risk stratify your patients in GI clinic, for example, based on that data. All right. And so with that, I'll just summarize to say that this is where we see the initial use case for this technology, specifically the functional T-cell assay. A child will present with symptoms of EOE. They'll then undergo a preliminary endoscopy to make the diagnosis. We envision not just one test, but a panel of testing. Currently, we're working on soy, wheat, and egg to complement milk. We'd like to expand that to 10 or 20 of the major foods. The clinician would then use that information to design a custom elimination diet, and 6 weeks later, confirm remission via second endoscopy. So theoretically, we're moving the diagnostic process from diagnosis to remission from years, in some cases, down to potentially several months. So as I mentioned initially, this EOE milk test is going to be available at CHOP through the clinical immunology lab. We do need to determine, we need additional data on the performance characteristics of the other tests that we're developing. We do want to make this broadly available. CHOP is not going to be able to be the reference lab for this testing. I think the demand is going to quickly make that not feasible. So we're working right now with a national laboratory to try to move this technology and make it broadly available. And then, of course, I want to spend the next several years determining more food peptides, more HLA molecules, and more TCRs that are mediating this disease, because I really think that that's going to be foundational information, not only for the next generation of diagnostics, but also potentially for the next generation of therapeutics for EOE and food allergy more broadly. So with that, I would like to just make a few acknowledgements. Again, I'm very thankful to the department for the invitation to speak today. I need to acknowledge both Julie and Cleo, who are boxed there, did the majority of this work. Several collaborators of note, but in particular, Jonathan Spergel, the head of my division, who's been a close collaborator for many years on this work, as well as Michelle Passler, who is the head of our clinical immunology lab. I can't complete without thanking our CRCs, of which we've had many who have done the tireless work of identifying and recruiting these patients. And we're very thankful. And then, of course, my funding sources. And so with that, I'll say thank you very much, and I'd be happy to take any questions.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.